Hello again everyone, welcome back to ptet.chemistry channel. So in this continuation of rate of reactions, this is for the combined science or the basic elementary science as part of the combined science syllabus for the O Level or the GCSE or IGCSE combined science syllabus. Anyway, so we're going to be talking about the effect of surface area. So surface area means what comes into contact together between the reactants. So when you have a big lumpy thing there and then you put in a solution, so only the surface, only the outer surface is going to come into contact with your solution. So that means the thing which are inside here is not going to come into contact with the outside here. Whereas when you crush it into fine powder, so I don't want that. I want to say that when you crush it into fine powder, let's say you have smaller pieces, then you have increased the surface area because you have crushed it into finer powder. So each of these are in contact. Each of these are in contact with the solution, and whatever that comes into contact with the solutions, they can react together if they are meant to react. So there are larger surface area. They are going to react much faster. So larger surface area for the reacting particles, your rate is gonna be higher. Your rate is going to be higher. Okay. So basically, the smaller the size of the reacting particles, if you can crush up your big reacting particle into smaller area, smaller surface. If you crush it into smaller size, then you will have greater total surface area because what comes into contact exposed for reactions is going to be higher, and therefore you get faster rate of reaction. These are your descriptions, very, very important. To talk about surface area doesn't mean that the smaller the size, the lower is the surface area. This is the one that a lot of students always get confused about. Because when you crush up something that big, you are exposing more of the reacting particles to the other reactants. And when you expose it to the other reactant, you increase the total surface area. Larger surface area comes with smaller size of reacting particle. Now this is calcium carbonate. This H plus is basically acid. So you also know this from year 9 chemistry content that you have a carbonate. Carbonate plus acid, you will get a salt, and then you will get carbon dioxide gas, and you will get liquid water. Carbon dioxide is a gas. As you know, CO2, this is a line diagram. It is not dot and cross, but then your year 9 chemistry covalent bonding topic likes to ask you to draw the dot and cross where you need to complete the outer shell. They're all part of the syllabus as your case study there. And of course, there's water as well. So H2O, so hydrogens dot cross oxygens dot cross oxygens, and then 2 dot on the oxygen because oxygen has 6 outer shell electrons. From your periodic table, group 6 element has 6 outer shell electron. Now calcium carbonate is also marble chips. So what we have is we will keep this mass of marble chip, concentration of acid and temperature are kept constant. These are the kind of things they like to ask you in a practical, alternate to practical or a practical investigation. Why do you keep them constant? That's the thing. You keep them constant because when you change the mass of marble chip, you can affect the amount of gas being given out. You can affect the rate of reaction. Changing any of these things here, which you want to keep constant, they can affect the rate of reaction. That's why you keep it constant. Constant means not changing. If you do not want it to change, because it is not what you want to investigate. As you want to investigate surface area here, you keep all the other factors that could affect rate of reaction constant. That is what you call control variables. I briefly talk about this towards the end of the last lecture tutorial when I talk about concentrations. So control variables are the things that could affect your reading, your rate of reactions, but then you keep them constant here because you don't want to investigate changing these factors. Now what happens is you have lumpy form, lumpy meaning like a big piece here, or you have smaller lumps where you crush them into finer powder like this thing here. The rate of reaction may be followed. Measure the time taken. So here, this is what you measure. Measure the time taken to collect a specific volume of gas. A specific volume of gas. So this is just a specific volume of gas. But what are you measuring? You are measuring time. To measure time, you need something called stopwatch in the lab. This one is a primary school mathematics, even not primary school mathematics actually, primary school science. Even primary school students would know what a stopwatch is to measure time. Even lower secondary science students would know what a stopwatch is to measure time. So this is what you measure. What you measure is going to be what you call independent variable. So that is what you call independent variable, because question can ask you identify or state the independent variable. Oops, I beg your pardon, sorry. What you measure is dependent variable, because what you measure is the y-axis. Remember I talked about in the last lecture tutorial, why are you so dependent? Why are you so dependent? That is what you measure. On the other hand, what you change is your x-axis. So what you change is going to be your independent variable. Your x-axis is your independent variable. Your y-axis is your dependent variable. You change the x, you measure the y, you keep all these other things constant. They are called control variables. As I said, the rate of reaction is going to be faster with the larger surface area. As a result of the smaller size of the reacting particles, you expose more of the reacting particles for reaction, and therefore you get a faster reaction. In general, the greater the surface area, the faster is the rate of reaction. Just don't get confused. When your particle size is smaller, you have actually greater or larger total surface area exposed for reaction. Therefore, you have faster reaction there. Okay? Now the next bit is about temperature, effect of temperature on rate. In most cases, as you could see here, as well as here, as temperature increases, what happens to the rate? Rate increases as well. Here, this is an enzyme catalyzed reaction. This is in combination with your biology. This is called enzyme catalyzed reactions. What you have there is, you have this optimum temperature. Optimum means is the best temperature for that particular reaction. As you can see, there's the highest rate. This is the highest rate, is the fastest rate at this particular temperature. We know that enzymes are present in biological bodies because at very high temperature, what happens to enzyme is that enzymes get denatured. That one you have to ask your biology teacher to explain more to you. So enzyme will change its shape and therefore they lose what they're supposed to do, which is as a biological catalyst to speed up the rate of reaction in your body. Okay, so they get denatured at high temperature, they lose their shape, they can no longer do enzyme catalyzed reactions, your rate slows down there. Okay, so this is just an example of a reaction. What is important is there are stat symbol here. So this is the sulfur solid. We started from aqueous and aqueous. Aqueous and aqueous means you have solution. This is again year 9 chemistry. Under stat symbols, solution means can see through. That means it's transparent. Transparent, you can see through it. This is a precipitate or a solid. Cannot see through because a solid is opaque. Opaque is a physics properties. You have learned these probably in lower secondary or primary school science. Opaque, you cannot see through it. So we mix these two together and we will see the time for these solutions to go opaque when the solid appears. When the solid appears, this cross will disappear because you cannot see through it anymore, whereas previously at the beginning you could see through it. So the rate of reaction may be followed. Measure the time. Measure the time taken for the precipitate. Precipitate meaning to say your solid in order to be formed. So that is the thing to be measured. What you measure. Remember, what you measure is on the y-axis. This is on the y-axis. Why are you so dependent? Why are you so dependent? So the y-axis is what you measure. What you measure is called dependent variable. Now we need to keep a couple of other things constant because we know that volume and concentration of both the reactants. So here, this is an example of these two reactants. They've just said concentration of both of these. But if you have a different reaction, then you just say concentration of your reactants. Concentration of reactants based on the question. Okay, you keep them constant. You keep them constant because we are changing temperature. So this is what you change. we are interested in investigating the effect of temperature on the rate. So what you change here is on the x-axis, and what is on the x-axis is what you change. That is what you call independent variable. Independent variable is what you change in the experiment, is your x-axis, and you measure the time taken for that solid to appear per the balanced chemical equation here. This is the y-axis dependent variable. These things are kept constant. So the thing that you keep constant in this kind of experiment, those are called control variable. Control variables are the variables that could affect your rate of reaction, that could affect what you're trying to measure. That's why we don't change them, because we are not interested in changing this. We are interested in changing temperature for this particular experiment. Well, what they found is the time taken is shorter when you perform it at a higher temperature. So the higher the temperature, the faster is the rate of reaction. That means you see the precipitate faster, you see the solid form faster. That means the thing become opaque much faster. That means the cross disappear much faster. Therefore, the time taken is much shorter. Be careful about rate and time. So if the time taken for the reaction is shorter, your rate is going to be higher. That means your rate of reaction is going to be faster. Higher temperature, faster rate of reaction. That is the takeaway message there. Last but not least, we need to talk about catalyst as stated in the syllabus. So definition of catalyst, they can ask you, what is a catalyst? A catalyst is a substance which increases. So this thing here, alter, you can change it to increases the rate of reaction. So it makes the reaction go faster, therefore it increases the rate of reaction, but it is left chemically unchanged. It does not change itself chemically. Its identity does not get changed because it does not get, it does not change at the end of the reaction, but it could be involved in the reaction. That's why it's a catalyst. Okay, so what is very important is you don't need a large amount, you only require in a small amount. You use it to speed up reactions, to increase the rate of reactions. Example, you have enzymes. Enzymes are biological catalysts. Biological because we use enzyme in our body. Animals or plants, they use enzymes for certain biological activity. So it's all living things, it's natural kind of catalyst. So that is your enzyme. In industry, a lot of chemical reaction might take place very slowly, and therefore we use not enzymes, we use other catalysts in order to speed up rate of reaction. That's the main purpose of a catalyst. So just to summarize what we have done from the first lecture tutorials, we have described the effect on the rate of reactions. Concentration of solutions, the more concentrated you are, the more particles you have in the same volume, they can collide more frequently, and therefore the higher the concentration, the faster is the rate of reaction. Now the second bit was on surface area. The smaller the surface area, the greater the surface area, that is when you crush up a big piece into smaller or finer particles, you expose more of the reacting particles together, and therefore you actually increase the surface area as you grind them up into smaller pieces, and therefore you will get faster rate of reaction when you have smaller particles because they have greater surface area, they are exposed for the reaction. Be able to explain as well. In terms of temperature, in general, when temperature increases, rate of reaction will increase because particles will have more energy. When you increase temperature, we talk about it in year 9. Year 9 heating curve, when you increase temperature, your particles will gain more kinetic energy, and therefore they collide more frequently, and therefore they give you faster rate of reaction. This is true for most reactions, also for enzyme reactions, but what is special about enzyme catalyzed reaction is that it's to do with biology, so it's a biological catalyst, so that means they don't work at very high temperature. Just like that, you know your body cannot work at high temperature above 40 degrees Celsius, that's why fever is very dangerous for us human beings. In general, the higher the temperature, the faster is the rate of reaction. Last but not least, we have the influence of catalyst. It's basically a substance that will not change chemically, so it's chemically unchanged at the end of reaction. But what it does is it increases rate of reaction, it makes reaction go faster. We already seen enzymes as a biological catalyst. In industry, in real life, we have many other non-biological catalysts that will help to increase the rate of reaction. That's why we use them in real life industry. All right, thank you for watching, and I'll see you and explain more on rate of reactions in the next series of the lecture tutorials. All right, don't forget to click the button on the bottom right to subscribe to my channel. Follow me at ptet.chemistry, that's at ptet.chemistry on Facebook, Instagram, Twitter, and Telegram to get connected.